So you're probably all glad right now that you're not taking an exam, right?
Yes.
Yes.
But tomorrow at 9 you're going to go.
Oh, he's going to review today in class.
No. I'll be in my office if you have any questions. Wednesday, there's no new work on your unknown, although you should consider maybe on Wednesday making a fresh slant of your unknown, just probably time to subculture it if you haven't done it in a while. We'll be doing a lab about counting microorganisms, and there will be some opportunity for optional points depending upon the results that you get. You'll be working in pairs. And then I will also go over expectations for the lab exam, because you do have the lab exam the Wednesday that you come back from break. Okay, so most of the lab exam stuff falls into two categories, and that is stuff you are routinely doing. You just have to demonstrate you can do it. So you will have to do a gram stain, for example, from memory. You'll have to do a streak plate for isolation. You'll have to do a hanging drop slide in Showman. You'll have to be able to calibrate a microscope with an ocular micrometer and a sage micrometer and measure some stuff. The other big thing is review questions at the end of each lab. Those are good ideas of kind of short answer questions. But we'll go over kind of general expectations, and I'll tell you some things you need to know and some things you don't need to worry about quite as much. So we'll do that Wednesday.
The optional points questions you sent for those videos, are those due tomorrow?
No, they're due.
Later.
They're due by the end of the semester. You can do those anytime by the end of the semester, and there will be another big bank of things once we have the first lab exam called ID Bactors, and those will be due at the end of the semester too. So, yeah.
Those are best in biology videos, right?
No, they're from a series called Unseen Life on Earth, and they're DVDs that Natalie has.
I think I googled it. I think you can watch them online too.
Can you watch them online?
I'll be able to.
Okay. Was it on YouTube or?
I just typed it in on Google and the website popped up, and they just have like a little.
Oh, okay. I didn't realize that. That's nice.
Thanks, Dr.
Okay, so we're in a new topic now, so don't worry about this stuff being on the exam, at least this exam. Did I tell you same format for exam tomorrow that previous exam, except you have all 3 essay questions, not just 2? All right, so we're talking now about chemotherapeutic agents. What's the big distinction between the chemotherapeutic agents and the general chemicals that were disinfectants and antiseptics that are on this exam? Big difference.
Chemotherapeutics have to be a lot more specific.
Yep. Highly selectively toxic, okay, because the antiseptics and disinfectants are disinfectants designed for use on the body? Nope, they're for inanimate objects. Antiseptics are for external use only. You kill a few skin cells, no big deal. Okay. You start taking something internally, and then you need that selective toxicity. I'd like to lead into this discussion a little bit by doing a little bit of history. I really like history of science. I think that you can learn a lot about kind of where we've been and how far we've come by looking at history of science. When you start looking at the history of science in regard to chemotherapeutics, the first strategies that were employed were not really strategies to find these highly selective toxic agents. Instead, the earliest strategies that were used to try to prevent and treat disease were things that would enhance the body's own natural killing mechanisms, which are designed to kill infectious agents, i.e., enhance your immune system. This dates back to really the mid-1700s. And the name that's probably most well associated with these early efforts of enhancing immunity is Edward Jenner, who was a British physician. Anybody know what Jenner's famous for? What kind of procedures am I talking about here? Something that you can do that will enhance your immunity, make you more potentially resistant to getting a disease. Immunization, okay? Jenner developed the first immunization. You know what it was against? What disease? Smallpox, okay? Smallpox, which has now really officially been eradicated. It is one of the few diseases that has been eradicated. There are a few samples still left in guarded government facilities for research purposes, but smallpox, which used to be a scourge, has now effectively been eliminated by immunization. Smallpox is caused by a virus, the vaccinium virus. That's actually where the word vaccination comes from, was this particular method that was used to try to vaccinate against this particular disease. And one of the things that Jenner noticed in his observations was that milkmaids, these were British women who milked most of the cows, did not get smallpox as often as the general population. They did, however, get a milder form of a related disease called cowpox. And they would pick that up from handling the udders and milking the cow. But they got a little bit sick from this disease, probably a little fever, maybe a little rash, but it wasn't lethal like smallpox was. But then when the smallpox epidemic came through town, these individuals were more or less immune. So his strategy was, let's try taking some of the material from lesions from somebody who has cowpox, because it causes, kind of crusty lesions, and inoculate that into a healthy individual and see if it can confer some immunity. And it did. And you could actually do just kind of a little scratch thing where you took a little needle, for example, and scraped away a little bit of the cowpox lesion, and then scraped somebody else's healthy arm with that, transferred some of those virus, and what's happening? Well, the body's immune system is recognizing the cowpox virus and mounting an antibody response against it. And there turns out to be a cross-reactivity between cowpox virus and the smallpox virus, and those antibodies conferred some immunity. So that was the first really recognized and published immunization protocol. Other people in the 1800s expanded on this particular technique. Pasteur developed a vaccine. Louis Pasteur had his hands in pretty much everything. You may know what vaccine Pasteur developed? I remember my kids reading a little golden book in school about Louis Pasteur and his rabies vaccine. He was the first person to develop a rabies vaccine. In the early 1900s, sexually transmitted diseases like syphilis really stimulated a search for a selective agent that would kill this particular pathogen and leave the host relatively unharmed. Probably one of the most famous people that spearheaded these particular types of efforts was Paul Ehrlich and his colleague Hatta. Ehrlich really spent his life on a search for what he liked to call a magic bullet. And the idea behind the magic bullet was this selective toxicity, a drug that could target the pathogen but not harm the host. And in particularly, he was interested in a magic bullet against the sexually transmitted disease syphilis, because that was really, again, a widespread disease, no treatment for it, and it was really a societal scourge. And when we talked about the survey of the microbial kingdom, I showed you the primary, secondary, tertiary syphilises, and it can be lethal. It's kind of an interesting disease. You'll get an outbreak and then it'll go dormant. And then you'll get a re-outbreak having a secondary disease, and then it'll get dormant again. And if it's untreated, eventually it will be lethal. Ehrlich and Hatta in, I think it was 1910, early 1900s, developed the first selective agent to treat syphilis called salvarsan, and I think I've mentioned this before. Salve meaning that it supposedly gave you salvation from this particular disease. And what is the AR? What kind of compound? This is an arsenic-based compound. Okay? And salvarsan killed syphilis bacteria, but it wasn't completely selective in its toxicity, and therefore it also had some horrible side effects. Many people argued that the cure was worse than the disease. But this kind of idea and this concept of the magic bullet was in everybody's mind. And as we started to learn more things about the characteristics of pathogens and how they differed from the host, basic concepts like pathogens are prokaryotic, hosts are eukaryotic, really led to some interesting ideas and strategies about how to develop these selectively toxic agents. A real breakthrough came in the 1930s. A German researcher by the name of Domack was studying the ability of various organic dyes to kill microorganisms. And he was working with one organic dye, it was a red dye, called prontosol, and saw that it had some interesting antimicrobial properties. If you exposed bacteria growing on a petri plate, for example, to media containing prontosol, it had very effective inhibition. However, then, when you took it to the next step and he tried to do some treatment with living animals, prontosol was a flop. It didn't work at all in vivo. So, for example, if you had a dog with an infection, you gave it this dye. Prontosol didn't help at all. Well, in the early 1930s, his young daughter got a very innocuous finger prick. And that finger prick turned into a septic infection. And septicemia at that particular time was pretty much a death sentence. So he was pretty desperate. So what he tried doing was he tried injecting his daughter with prontosol. And this wasn't just necessarily a Hail Mary last hope. Because he had done a lot of work with animals, and one of the things that he realized is when you put a drug in an organism, many times that drug is metabolized and is turned into some other product. And prontosol, as it turns out, in humans, is metabolized into a different compound called sulfanilamide. And sulfanilamide had very, very good antimicrobial properties. And as it turns out, this particular drug saved his daughter. As it turns out, this was one of the very first recognized magic bullets, and its function is that it worked by competitively inhibiting a key bacterial process that humans didn't have. And we'll talk about that key bacterial process in just a second. Sulfanilamide was the first of a whole generation of sulfa drugs. And we still have some sulfa drugs that are in use today. Sulfa drugs were kind of the precursors to antibiotics. Has anybody ever seen old like World War II films where there are World War II scenes and someone who's injured in the battlefield, they've got these metal containers and they sprinkle this powder in the wound? Those are sulfa drugs. Okay? They're not antibiotics. They are these kind of competitive inhibiting compounds that are called antimetabolites. Let's take a look at sulfanilamide for a second to kind of get this idea of how these antimetabolites work. In human metabolism, there are a number of coenzymes which are absolutely important for proper enzyme function. Okay? Coenzyme A, for example, in cellular respiration. An important coenzyme in cellular metabolism is THF, tetrahydrofolic acid. THF is an important coenzyme in a biosynthetic pathway that leads to the synthesis of both purine and pyrimidine nucleotides. And if you don't have purine and pyrimidine nucleotides, you can't make new DNA and RNA. That's a problem. Well, there's actually a fairly long biochemical pathway that results in the synthesis of this coenzyme, THF. It starts out with a precursor molecule called PABA, para-aminobenzoic acid. PABA is converted by an enzyme into dihydrofolic acid. Another enzyme converts that into tetrahydrofolic acid. And then this particular coenzyme works with other enzymes in the synthesis of purine and pyrimidines, which are necessary to make DNA and RNA. Okay? So, the enzyme that catalyzes this particular reaction has an active site that is complementary to the substrate PABA. Okay? And if the enzyme binds to that properly, then the catalytic reaction takes place and forms a dihydrofolic acid. And then another enzyme, etc. Sulfanilamide and some of the other sulfa drugs have a very similar structure to PABA. And this particular drug competitively inhibits this enzyme, which normally converts PABA into dihydrofolic acid. It competitively blocks this particular enzyme. And therefore interferes with folic acid synthesis, and therefore DNA and RNA synthesis. So how can this be a magic bullet? Okay, how can this be selective for pathogens and not for humans? Well, we don't have this particular pathway. Where do we get our folic acid? We get it in our diet. Okay? This is an essential nutrient that we cannot make. Green leafy vegetables are high in folic acid. So when mom says eat your vegetables, there's a reason. You need to get this stuff in your diet. Bacteria synthesize their own folic acid. And therefore, they have this enzyme that we don't. And that enzyme can be competitively inhibited. Now, there is an exception to that particular rule. There is one bacterium that has to synthesize, or I'm sorry, that does not have to synthesize its own folic acid. And that is the causative agent of tuberculosis, Mycobacterium tuberculosis. And therefore, sulfa drugs are not effective on TB infections. There's some other drugs that, other sulfa drugs that block other enzymes in this particular pathway. There is a sulfa drug that is used fairly commonly called Septra, and it is used in treating bladder infections. And it's pretty effective in this particular, blocking this particular pathway. The idea of competitive inhibition and shutting down a key metabolic pathway in the pathogen is a really powerful tool. I was just reading the other day, and one of the real public health threats that's re-emerging right now is multi-drug resistance tuberculosis. These are strains of tuberculosis that are basically resistant to all known forms of antibiotics that we have today. What's so scary about this is that there are about 30% of people on this planet who are carriers of multi-drug resistant tuberculosis. They might be asymptomatic. But if you transmit that particular pathogen to a susceptible individual, oftentimes a person with lower immunity, you've got a real problem. There's a new antibiotic for multi-drug resistant tuberculosis. It was just approved by the FDA in 2013. Production of new antibiotics, we'll talk about this later, is really plummeting because there's not a high profit margin in many of these, and the approval and regulatory process for getting these drugs approved is really lengthy. I would bet that the first studies on bedaquiline probably started in the 90s, probably sometime. It takes decades sometimes for the approval process to take place. This particular drug selectively targets a bacterial ATPase. And evidently there are some small but significant differences in ATPase in our cells and bacterial ATPases. Remember what an ATPase is? That is that proton pump in the inhibitor, in the membrane. And as protons flow through that proton pump, you can make ATP by chemiosmosis. Okay. So a selective inhibition of this particular pump. This really points out that fundamental knowledge of metabolism is really important if you're going to identify and design drugs to try to inhibit key aspects of those metabolic reactions. So the people who worked out this original pathway of synthesis of tetrahydrofolic acid weren't doing it because they were trying to discover a drug, but that basic research of knowing how metabolism works then gives clever people insights about how can I go about then exploiting some of that knowledge and using it to identify or design drugs to, in this case, critically inhibit it at certain particular steps. Okay? How would we, really start looking for an ATPase synthase inhibitor if we didn't know how the electron transport chain worked and how chemiosmosis worked? So this whole idea of basic research being the foundation for applied research is something that's really important. And it's something that Congress, most people in the federal government that are responsible for funding, and the general public really do not understand and appreciate. It's the foundational work that's done by investigation for, at least initially, knowledge's sake, and just trying to figure out how something works. But if we don't have that bedrock foundation, then the applications, we really can't develop. Okay, here's a very interesting guy who had a pretty amazing impact on a number of people. And this is Alexander Fleming. This particular picture was probably around 1928. Fleming looking at Petri plates under his microscope. And probably one of the things that he saw when he was looking at that was this particular fungus. Or maybe he saw Petri plates where there were fungi growing on Petri plates seeded with a bacterium, maybe staph bacteria, and noticing these particular zones of inhibition that sometimes occur around fungal or other bacterial colonies. Fleming is actually credited with, although some people will disagree, the discovery of antibiotics. Most renditions you read about Fleming indicate that the discovery was really an accident. He just happened to have some contaminated Petri plates that had a lot of fungus on them. You've all had those before. But before he just tossed them away, he was working with glass Petri dishes, and so he would have to wash them first and dispose of the media. And he started to look at it, and he said, wow, there's some fungi growing on these plates that are inhibiting microbial growth. And we thought it was kind of interesting. But really then, nothing much happened with it. And this discovery of molecules produced by one organism that inhibit the growth of the other, which we call antibiotics now, against life, really was just kind of a curiosity. At least until World War II. And particularly in the Pacific portion of World War II. More soldiers were lost to battle wound infections than were lost to any other cause of death, particularly in the tropics. Hot, humid environments were wonderful for particularly fungal growths and also microbial growths, warm, humid conditions. And so even what looked like relatively minor wounds could become raging infections, and if sulfa drugs didn't work, then soldiers were dead. So this rediscovering of the inhibitory processes of antibiotics was really important, and some people really credit that, along with the nuclear bomb, of turning the tide of the war, particularly in the Pacific theater, because we had access to these drugs where the Japanese did not. Now, historically, there are anecdotal writings that indicate that this knowledge has been around for some period of time, using molds to treat diseases. There are references in ancient Chinese writings of applying moldy bread to wounds that are infected, and that those would have then some kind of curative power. Well, that's probably because the molds were antibiotic-producing strains of fungi, like this Penicillium notatum fungi, which was the one that Fleming was working with. Antibiotics turned out to be the true magic bullets. And the impact of the discovery and production of these particular drugs was nothing short of miraculous. There was a similar discovery in the insect world of a super chemical called DDT, which again, around the time of World War II, was seen as just this incredible discovery that would change the face of the world. One of the diseases that was very common in World War II was an arthropod disease transmitted by fleas called typhus. And I've seen old pictures before of when we drove the Germans out of Rome, for example, that when the Allies came in, they had these looked like big insect sprayers, and they were just spraying powder of DDT all over people to kill the typhus-bearing fleas that they were infected with. And DDT was thought to be this miraculous drug. Really, entomologists thought at one point in time DDT would be able to eradicate all insects. And that was actually a goal. All insects were bad, and so we should get rid of all of them. So what happened with DDT? Well, we started discovering some toxic effects, but we also discovered that very quickly insects were developing resistance to DDT. What happened with antibiotics? As a result of overuse, which we'll talk about later on, organisms are now developing significant resistance to antibiotics. And we are literally in a war with bacteria, trying to develop chemicals that will selectively kill them. And while we're doing that, bacteria are evolving resistance to those particular chemicals. The chemicals, the antibiotics, are actually the selective agents that are driving the evolution of the resistance. It's really a very interesting back and forth that's going on right now. As far as antibiotics are concerned, there are two major classes of antibiotics. There's actually more than two, but there's two big classes of antibiotics. A big group of antibiotics are cell wall inhibitors. And those include penicillins and cephalosporins. These particular antibiotics all have a similar structural theme, and we'll look at that. But these are called the beta-lactam antibiotics. And probably most of you who have ever taken prescribed antibiotics have probably taken one of these, either penicillins or cephalosporins. The other major class of antibiotics are the protein synthesis inhibitors. We'll talk about some of those as well. Probably you've had less likelihood that you've taken one of these, although some of you may have had erythromycin, for example. Tetracycline was one of the early protein synthesis inhibiting antibiotics. Well, if you look at these two categories, they represent some very low-hanging fruit in terms of exploiting differences between pathogen and host that can give you selective toxicity. For example, cell walls. We don't have them. So if you inhibit cell wall formation, obviously you've got a way to selectively kill the bacteria. For protein synthesis, we have protein synthesis, but again, 70S ribosomes versus 80S ribosomes. Our cytoplasmic ribosomes are 80S. Our mitochondria, unfortunately, are 70S. But this gives us, again, a way to exploit a key difference between the pathogen and the host. The early antibiotics that were developed, including the naturally occurring form of penicillin, penicillin G, were primarily effective only against gram-positive bacteria. The reason that penicillin was not effective against gram-negative infections was that LPS layer that the gram-negative bacteria. The structure of penicillin is such that it's not easily transported across that gram-negative LPS layer. Gram-positive lacking it, it's quite effective. So one of the things that were done quite early on were to develop some derivatives of penicillin. Organic chemists started tinkering around with the molecule and started substituting some functional groups here and there and creating some new novel penicillin derivatives, and these were called semi-synthetic antibiotics. Technically speaking, the term antibiotic is only supposed to be used for a naturally occurring compound produced by metabolism of either bacteria or fungi. The ones that are fine-tuned in the lab are technically supposed to be called semi-synthetic antibiotics, but we just lump those all together and call them antibiotics. This shows you the sources of some of the common antibiotics and semi-synthetic antibiotics. What you'll find is that there is really a kind of two groups of organisms that typically produce those, and there are some fungi, including penicillium fungi. Most of the bacteria that produce antibiotics are gram-positive rods. And the two most common genera are the genus Bacillus and the genus Streptomyces. Particularly Streptomyces, if you look down here, there are several different species of Streptomyces that produce several different types of antibiotics. These are common soil bacteria. Now, there are a couple of gram-negatives that produce antibiotics as well. Pseudomonas fluorescens is a gram-negative bacterium. But by far, the vast majority of them are produced by gram-positive bacteria. Okay, they're produced by metabolism. And specifically, fermentation. What would be the logical reason you would assume that organisms, particularly soil microorganisms, would produce by fermentation these molecules that are called antibiotics? I mean, it's costly to do this. Metabolically, it's costly. There must be a selective advantage of doing this when you do it. What would be the advantage? You're a bacteria living in the soil. Yeah, I mean, you can basically, the idea was kind of conduct chemical warfare on your neighbors, because the soil is a very complex environment with all kinds of different organisms competing for common resources. So if you can kill your neighbors, you gain a competitive advantage. And that has been kind of just the common wisdom pretty much as long as we've known and appreciated antibiotics. But that wisdom's actually starting to be questioned now, because they've done some interesting levels of measuring actual concentration of antibiotics that these bacteria can secrete into the soil, and they're very, very low. And many people argue that the concentrations are low enough that they really don't have any effect on your neighbors. So what's going on there? Well, there are all kinds of alternative hypotheses that have been floated, and we're still trying to sort some of those out. Some people feel like that they're just some kind of metabolic curiosity. They're kind of a vestige of some form of metabolism that was tried by a group of microorganisms, and it just kind of retained, kind of like vestigial structures. Other people think that maybe antibiotics serve signaling roles, particularly in complex biofilms, and that their ability to kill other organisms in vivo is just kind of a coincidence. But this idea of long-held beliefs then becoming questioned, I think is an important idea for everybody to understand. And the idea that we always thought that this is just a chemical warfare agent is now being questioned by at least some individuals. The cell wall inhibitors work by interfering with the resynthesis of peptidoglycan. Remember that in order for bacteria to grow, that cell wall has got to be selectively loosened to accommodate water uptake and swelling of the cells, because uptake of water and cell swelling is what causes bacteria to increase in size and grow. But then you can't just have that cell wall loosened forever. It's got to be selectively weakened and then resynthesized, weakened and then resynthesized. So if you interfere with the resynthesis of the cell wall, that can cause a weakened cell wall, and then in hypotonic environments, those cells can lyse. Okay? So on the right-hand side over here, we have a picture of some normal cells in the absence of antibiotic. And then these are cells that have been grown in the presence of antibiotic. And you can see that a number of those cells have now lysed. Okay? As the cell wall has become weaker. Well, here's another instance where basic knowledge is really important in development strategies for making new antibiotics. If this class of chemical compounds interferes with cell wall resynthesis, knowing something about how cell walls are put together is really pretty critical. Because then you can start to say, gee, maybe I could exploit this step to inhibit, or maybe I could exploit this step to inhibit. So, here's something I just threw together this morning. This is not in the current version that's on Dropbox, but I will update Dropbox to include these particular slides. So let's do a little review of cell wall structure. Okay, cell wall consists of both gram-positive and gram-negative peptidoglycan as the main structural component. It's by far the main component in gram-positives. In gram-negatives, you have less peptidoglycan, but you have that significant LPS layer. But we're concentrating just on the peptidoglycan right now. Peptidoglycan basically consists of these long polymers of these two sugars, NAG and NAM. Remember those? And then peptidoglycan chains can be bonded together by these peptide inner bridges. The sugars, NAM, have these 5 amino acids attached to them, forming a tetrapeptide NAM structure. So this is kind of the basic structure of peptidoglycan. So how's this made? Well, initially, peptidoglycan starts being produced in the cytoplasm of the bacterial cell. And there's a couple things going on. There are enzymes that catalyze a reaction where this sugar NAM has 3 amino acids added to it. And then at the same time, there's a different enzyme that hooks together 2 alanines, 1 L and 1 D. And so this 2 alanines and 3 amino acids join together to form this NAM pentapeptide. It's a sugar with 5 amino acids hooked onto it. Well, there's an antibiotic called cycloserine which inhibits this particular step of hooking together those 2 alanines. If you don't hook together those 2 alanines, you don't make the NAM pentapeptide, and that's a key component of peptidoglycan. Okay, so you can inhibit its resynthesis at this particular step right here. This NAM pentapeptide attaches to a carrier. The carrier is called a bactroprenol. And that carries that pentapeptide across the membrane after it adds the second sugar to it, NAG. So now we've got the NAG, NAM, and that's got the pentapeptide. Okay, so that's over here. Well, there are 2 enzymes that work to take this NAG, NAM pentapeptide and hook it onto an existing peptidoglycan chain to lengthen it. There's an enzyme called transglycosylase that pops these 2 things and joins them to the chain. And then there's an enzyme called transpeptidase that hooks these pentapeptide chains together to give them cross-linking strength. Okay, so there's a couple of key enzymes here that we can inhibit. All of the beta-lactam antibodies interfere with these 2 steps right here. Okay, they interact with either transpeptidase or transglycosylase. So penicillin, any of the cephalosporins, all block at that particular place. Once you've knocked off these NAG, NAM pentapeptides, the carrier normally moves back across the membrane to pick up more. The antibiotic bacitracin interferes with the transport of this carrier back across the membrane. So you see how understanding the metabolism and the mechanism by which cell walls are synthesized, you can start looking at this and going, wow, there are some points of exploitation there, particularly key enzymes. And if you understand what the substrate for those enzymes look like, a clever organic chemist might be able to either make, identify, or tweak a naturally occurring molecule to have a similar structure to the substrate, and therefore competitively inhibit the enzyme. So penicillins competitively inhibit a couple of key enzymes involved in cell wall synthesis. So this particular slide just gives you some of the same kind of information in a little bit less detail, but again shows you the inhibition that takes place between the cross-linking, particularly of these sugars in peptidoglycan, and the resulting weakness in the cell walls that results that causes lysis of that particular cell. Naturally occurring penicillin, penicillin G, is a type of antibiotic that has a very unique structure It consists of this nitrogen-oxygen containing ring called the beta-lactam ring. And then attached to that beta-lactam ring is another ring structure called the thiazolidine ring. And that's the one on the right there. So all of the penicillins, whether they are naturally produced or semi-synthetic, have a beta-lactam nucleus attached to this thiazolidine ring. You can see how penicillin, this is basically the R group attached to this common structure, varies from one type of penicillin to another, or possibly even in this other class of antibiotics we'll talk about later, the cephalosporins. So methicillin, for example, is a semi-synthetic form of penicillin G. Organic chemists have modified this R group to be a little bit different. So, what's the purpose of these modifications? Well, there are several purposes, but some of the key ones are some of the modifications confer acid resistance to the penicillin molecules. Naturally occurring forms of penicillin are not acid resistant. So how do we have to administer them? By injection, right? You can't take a pill if the penicillin is going to be degraded by stomach acid. Okay? Another thing that the modifications are often targeted toward is increasing the spectrum of the drug. Spectrum refers to the range of types of organisms that the drug will inhibit. Naturally occurring penicillin G is kind of a moderate spectrum drug. It will only affect gram-positives because it can't get across that LPS layer. Some of the semi-synthetic forms of penicillin are more broad spectrum. They can get across that LPS layer and therefore are effective against gram-positive and gram-negative. The final set of modifications has to do with conferring resistance to penicillinase. What do you suppose penicillinase is? That is an enzyme that can break down penicillin. This is an enzyme that bacteria produce that basically can kill the drug that is intended to kill them. Methicillin, for example, this one right here, tends to be penicillinase resistant. Although, however, now Staph aureus have evolved a resistance to methicillin and have evolved the ability to produce a penicillinase enzyme that will now attack this particular molecule. So these are some types of antibiotics that you might have heard of before. Again, this beta-lactam nucleus consisting of the beta-lactam ring and this thiazolidine ring, and then the derivatives of this part of the naturally occurring molecule that occur in penicillin D, methicillin, ampicillin. There are various forms of synthetic forms of penicillin that have been produced. Amoxicillin, for example, probably most of you have had amoxicillin. Okay, so we're going to have a little break on this because obviously we've got the exam Tuesday, and then won't be until we come back from break and we'll pick back up with antibiotics. Word of explanation. Because the midterm grades are due tomorrow at noon, and I'm not going to have your exam graded, really it's just your first exam. However, I'm going to take a few liberties with midterm grades because 90 points out of 800 obviously isn't a midterm grade. So if you didn't do so well in the first exam, I'm going to bump you up a little bit based upon what I've seen in class particularly. I don't expect that your first exam is going to be your final grade. Okay, so don't sweat that too much. But if anybody challenges you or asks you about it, tell them this is really not a midterm grade.